Hello, listeners. Welcome to the latest edition of the podcast of the InnCoCells Project. And today I'm joined by Jaya. Jaya is our organiser here at the meeting in Luxembourg. And Jaya, I'll let you introduce yourself. You can tell our listeners what role you have in the InnCoCells Project and maybe a little bit about your where you work and what you do.
With pleasure. Hello, Richard. Hello, everybody. My name is Jaya Guerriero. I'm an Italian scientist. Now it's been more or less 10 years that I've been living in Luxembourg. So yeah, Richard, as you said, we are the host and we are very happy to have you all here in Luxembourg City.
It's been a really good meeting so far, I have to say.
So I'm a plant scientist and a plant molecular biology by training. So I did my studies in Italy at the University of Naples. And then I traveled around Europe during some postdoctoral positions. And then I ended up in Luxembourg in 2013, actually. And it has been very nice ever since working here in Luxembourg. And actually, we are really involved in our team with the study and exploitation of plant diversity, more specifically for plant molecular farming applications. So really valorizing the phytochemical diversity, but also the repertoire of other type of macromolecules of plants for different industrial applications.
So here in Luxembourg, you work at LIST, which is the Luxembourg Institute of Science and Technology.
That's correct, exactly.
Do you want to tell us a bit about your department and how many people are there and things like that? Yeah.
Absolutely.
Yeah.
So yes, indeed, I work at LIST, the Luxembourg Institute of Science and Technology. It's an RTO, a research and technology organization. We are around 600 people,
wow,
more or less. So we are quite a bunch of people spread across 4 departments. So I belong to the Environmental Research and Innovation Department, ERIN, it's the acronym. And more specifically to the Plant Molecular Farming Research Group, which is within the Environmental and Industrial Biotechnologies unit. So we have quite sort of architecture within our organization.
So it sounds like that's a perfect fit for the InnCoCells project. Very interesting, I think, for our listeners to learn how you got involved with the project. With your topical expertise, I presume you've worked with some of the partners before.
We have had some interactions with the coordinator, with VTT, a few years back. And we know, of course, the majority of the 17 partners involved in the project. We have been collaborating on and off along all these past years.
Have we met before in other projects? I can't recall.
No, I don't think so. I think that's the first time, actually.
Yeah.
I'm very happy to meet you. Absolutely. So how did we get involved in the project? Well, of course, we are working on a thematic that is very close with the subject of the project. So the expertises that we can offer in at least are very complementary to the, let's say, the ambitions of the project. So our role within the project is really in work package 3, which is on the upscaling phase of the project, where we will bring the lab optimized processes to a higher scale.
Interject there for our listeners. So work package 3 is the upscaling work package, and Jaya is the work package leader. So basically she's in charge of it and directs the progress and reports about it, etc. So she's really the best person to tell us about it.
With pleasure. So yes, indeed, VP3 is a central work package. It has a connection, of course, with the upstream work packages, which are, of course, involved in the determination of the bioactivity of the extracts or the mining of these extracts of the different plants and the biodiversity, and then optimizing lab scale processes in order to cultivate these plants, different systems. And then in work package 3, we actually inherit all the knowledge gathered in the previous work package, and we really try to bring the optimized lab scale processes to a higher scale, to the so-called pilot scale phase. And this is really important because this is a sort of pre-commercial stage that enables us to understand whether a specific bioprocess can meet the requirements of a full-scale commercial stage. And of course, we have still the time there to fine-tune and optimize specific parameters. So typically in this pilot scale phase, we study and we observe specific parameters like feasibility, cost, time, unpredicted results, and how to deal with that. Mm-hmm. So that's pretty much what we do in this work package.
So for the benefit of our listeners, when you say pilot scale, what sort of, are we talking a couple of litres or hundreds of litres or thousands? Yeah.
There's no specific actually definition of pilot scale. In this project, let's say pilot scale is everything that covers scales of 100 litres, more specifically between 300 and even cubic meter scale. But for work package 3, we have different unit of measures, if you want, because we have 4 different tasks within this work package. And we really try to compare and understand 4 different production systems for plants. So we have cell suspension cultures. So as the name tells, it's a sort of suspension of cells. So here we talk about liters, cubic meters, hundreds of liters. Hairy root cultures, which is pretty much the same volumes. Then we have whole plants, and these are grown in greenhouses, in aeroponics. And here we talk about surface, so square meters. And then we go up to the hectare in the fields, so real plants that are growing in the fields.
So again, for anybody who's interested, this idea of aeroponic cultures, we had a podcast recently with Frederick Borgard at Plant Advanced Technologies. Go and listen to that one because he goes into details about how the aeroponic system works. It's a very interesting podcast and I can't recommend it highly enough. So JS, back to your role in Work Package 3. Thank you very much for explaining the aims and objectives of that work package and what you're trying to achieve. Am I allowed to ask, I don't know, if I said how successful you've been so far, are you allowed to say which plants you've managed to, or which plant cells you've been able to grow?
I can provide a little bit an overview of the stage of the project.
That would be very interesting, I think. Yeah.
So now we are halfway within the project. We have 2 years ahead of us, and I would say everything is going pretty much as expected. Of course, we had to deal with some little hindrances, more specifically lab moves for some of the partners, but I think we dealt with that really well. There has been no long-term interruption. Everything has been running smoothly, so we have been able to deal with unforeseen events quite well. So I would say that so far we are pretty on track with the 4 different tasks that we have in this work package.
When you say unexpected, can you give us an example of something that's been overcome?
Sure.
A problem that's been overcome?
Well, something very practical. For example, at least we purchased a 300-liter working volume stir tank reactor, and there has been some delay in receiving it. Not too much, but we were supposed to start maybe our activities a little bit before, so we were a little bit delayed. But this did not have any negative impact on the execution of the different tasks, on delivering the different deliverables. So I must say I'm really happy with the advances of the work package so far.
Excellent. That's really, really good news. So maybe our listeners might not grasp the technicalities of this. So when you talk about scaling up a plant cell culture, you start off maybe with a litre of culture and you grow these cells in a flask which has a volume of 1 litre, and then the process of moving that to, let's say, 200, 300 litres. So on one hand you've got larger pieces of equipment, and on the other hand you've got more liquid biomass for the cells. So that sounds on the face of it like a fairly simple exercise. You just make more cells and put more liquid in this bigger vessel, but I get the impression it's not quite as simple as that. Just a brief description of the particular technical aspects which make it more difficult than just pouring more liquid into a bigger bottle. Yeah.
It may sound as simple as that, but I wish it was actually.
Yeah.
For sure, we have at lab scale optimized several parameters, so we already have a fairly good idea of how our cell suspension cultures, how they behave in a stir tank reactor. But of course, even if we apply exactly the same parameters in terms, for example, of temperature, light, for example, but above all, temperature is one of the key parameters, for example, or even how we agitate the cells. This is something quite important, because these cells, they are in a sort of dedifferentiated state with a thin cell wall around it. Mm-hmm. And even if the cell wall is an armor for the cells, there is still some, let's say, mechanical injury that we can infer to these cells when they are agitated in a reactor. So transferring those parameters from the lab scale, so let's say 1 liter, 2 liters, 3 liters, up to 30, 40, 100, 300, even cubic meter is challenging, because first of all, we need to deal with a bigger amount of growth medium. So we need to keep it sterile. So we don't need any contamination in it, because we just want our cells to grow in this culture. Mm-hmm. No fungi, no bacteria, no other type of living things, just our cells. And of course, we need also to make sure that the transfer of oxygen to the cells is optimal, so that our cells can be thriving in this environment, can perform, can grow, can divide, and then...
I see. So basically, the idea is if you've got a small bottle, air gets into it fairly evenly. But if you've got a massive volume of liquid, the middle part is long way from...
It's more challenging to ensure that every single part of the reactor gets pretty much the same type.
Which is why you need to stir it faster, and why it's then more stress on the cells. Yes, that makes sense. Yeah. So how do you overcome a problem like that?
Well, actually, it depends a little bit on the geometry of the agitation system that we have. So we may have the possibility to choose impellers, which are these systems that sort of blades on a shaft that agitate the reactor. We may choose some geometries of this impeller that will impact let's say, less negatively the growth of the cells. So, for example, the marine type impellers. And of course, we can choose to pump some oxygen in the growth in the reactor, so that some pure oxygen can be sent to the reactor. It really depends, but we have, of course, to, let's say, fine-tune also with the specific characteristic and behavior of the cells. For example, some cell cultures may tend to foam a lot. So if we agitate more, if we put more oxygen, then we may have this cake foam forming on the top of the reactor.
And what is the problem with that? What does that? How does that affect the cells negatively?
Well, if we have this foam, for example, in some of our reactors, the foam would go up and it can even go into the system, let's say, into the mechanical parts of the reactor, clog the filters, and so we may have some pressure problem.
So this all sounds very exciting with the success in scaling up so many different, not just different plant species, but also different formats. The cell cultures, the hairy roots, the aeroponic systems, and the plants being cultivated in the field and in the greenhouse. If you had to pick something like an ultimate achievement of what you'd like to get out of the project, something you'd really like to see work that you're working towards at the moment, what would you say that would be?
Well, there are several implications in the InnCoCells project. I'm not just talking about Work Package 3, but really the complexity and architecture of the whole project. What I would like to see is really that we manage to do something that can be useful for the, let's say, cosmetic applications. So the ultimate ambition of the project is really to find ingredients that can be put towards the market. What I would really like to see is that some of the species we are working on actually one day will be put on the market, some ingredients sourced from the species we are working on. And something that I really find very exciting in this work package and in the project in general is this interconnection between the different systems that we have. It's quite unique to compare different production systems. It's really nice to see that we can achieve something with plant cell cultures. The typical hosts for that are quite successful and quite robust are the microbial cells, but doing, trying to get phytochemicals or extracts from plants and cultivating them in large-scale bioreactors is very challenging and technically challenging. And so, let's say, together, I think we managed to find the optimal parameters depending on the species and how to upscale it up to the pre-commercial stage. So these are, let's say, the two things that I really would like to see by the end of the project, but I think we are on the right track to seeing them.
Well, with a full 2 years to still go, I think that does sound eminently achievable. Could this achievement be spread out further? Could you train others to undertake the same technology so they can take that forward?
Absolutely. Of course, LIS is an RTO, this is a research technology organization. We are really, looking towards making progress in different technological fields, but also transferring this knowledge. So the transfer of technology and the transfer of know-how is something that we cherish a lot. And I think it's also very important within InnCoCells to try to make a difference. And whatever we learn from Work Package 3 or from any other type of work package, that it can be somehow be useful for the community of researchers that are working on the same topic or interested around the same thematic. So in this sense, we discuss with InnCoCells partners, we discuss within my colleagues within LIST, and we decided that we would organize a vocational training revolving around the use of plant cell cultures as a safe means for the mass production of biomass for the extraction of phytochemicals with added value interest for industry. And so we are right now preparing this event in September. So
September this year. So a vocational training course for people interested in learning how to make plant cells and hairy roots grow at larger scales.
Exactly. Yeah. We will more specifically focus on plant cell cultures, but I think what is interesting is that we will really couple more academic lectures, let's say, in the morning sessions, and then we will have hands-on training in our labs at least so that the participants can really manipulate, set up a reactor and see with their own eyes and with their own hands what it means to run a reactor with plant cell cultures.
Have you done this type of vocational training before? Was it a new thing for InnCoCells?
It is something new for InnCoCells, this, let's say, hands-on training within the different participants of the consortium, and at least never so far organized a similar event. So it is the first time, and I hope it will be successful and interesting for the community so that maybe we can think about repeating it in the future.
Well, I'll tell you what, I hope that maybe we have an opportunity to do a podcast with some of the people who attend the course and get some direct feedback from them. I think that'd be a great idea. Yeah. Let me write that down in my diary. So we've talked a little bit there about the impact, if you like, of what LIST is doing, what your work is going to achieve for the project, for the wider scientific community. How about ending by talking about how this might work the other way around. What do you think the InnCoCells project will do for LIST? Will it influence your research in the future? Will it take it in new directions?
I think InnCoCells is a wonderful opportunity for our organization. It's a very articulated project, very fascinating, with very ambitious goals. Of course, LIST, by participating and by leading one of the work package, which is crucial for the execution of the project and gets a lot of visibility within the consortium, not only, but also among the stakeholder group. We have the possibility of showcasing our capabilities. So I think it's a win-win situation for the consortium and for LIST.
Fantastic. I've really enjoyed doing this podcast. Very upbeat.
Yeah.
Yes, it was a pleasure to do it. So thank you very much, Jay, for being a wonderful subject for the podcast of InnCoCells. And I'd like to end this by saying we're going to sign off now from lovely sunny Luxembourg. We're very much enjoying the 5th InnCoCells meeting. And please, listeners, tune in for the next edition of the podcast, which will also be from Luxembourg, and we will look forward to seeing you next time.